we have our immediate past president, Dr. Shamar BASAK, giving you a glimpse of his latest passion, which is genetics, and his deep diving into genetics of FECD. Yeah.
Very nice talk by Vikas and Padas and Shayan. So this is, I am taking a little interest in recently, so I'll be talking on genetics of Fuchs dystrophy in West Bengal, Eastern India. I do not have any financial disclosure. So if you see that even after 115 years, so we do not know exactly what is happening in Fuchs dystrophy. So there are so many, that's why so many studies are going on across the globe. It is a progressive disease. So we see in different form of Fuchs dystrophy in our clinics every day, starting from even with advanced Fuchs dystrophy and this kind of Fuchs dystrophy. But when you see this kind of picture in your slit lamp and retroillumination, the diagnosis is obvious, and specular microscopy is also important, confocal if you have, so Gattata and low cell count, pore morphology, pleomorphism, and polymegathism. There are two distinct clinical variants of Fuchs depending upon the age of onset, early onset and late onset. Early onset between 3 to 40 years, more severe but less frequent, we do not see much. What we see that is late onset, more than 40 years, which is perhaps less severe. So there are two ways of studies of gene in FECD. One is genetic linkage analysis of the family. Another is GOAS, that is genome-wide association study. So early onset diabetes, both are autosomal dominant and mostly have early onset mostly have a familial disposition, whereas late onset is sporadically familial. This is from IC3D classification. You see a lot of genes has been already identified. Although genetic factors are involved, the exact genetic role in FECD is not fully understood. It is likely that both genetic and environmental factor play a role. So what is known so far? FECD caused by mutation of a gene on multiple chromosome 1, 5, 7, 18 and X. Late onset FECD specifically, even in February there is one big review article appears in ophthalmology, the TCF4 gene on chromosome 18, and there is a trinucleotide repeat, cytosine, thymine and guanine CTG repeat. It is called CTG 18.1. If there is more than 50 repeats, it is significant for FECD. At the same time, it also implies the severity. If there are more repeats, severity of the disease process. There are other genes also identified, COL8A2, which is specially related to early onset FECD, and other genes are there. There are a lot of literature is there, and you can go to see this. So why genetic analysis in FECD is important in Bengal? If you see the data, primarily it happens, if you see the EK data, all EKs, 80% of all EKs in USAs are for FECD, whereas in South India it is between 10 to 17%. But in Bengal we have shown that earlier it was 22.5 in my DSEC era, and now in DMEC era it is 38.6%. Even in LV Prasad, the Bengali community of FECD patients who are going for EK, they are more than 25%. So the few studies available from India, from South India, Odisha, North India, and if you see that the TCF4 is between 17.3 to 34%. Then we collaborate with Niger, this is in Orissa, Bhubaneswar. So we used to send all the materials after doing DMEC or DSEC, and also blood. We call it FECD group. And also we control from non-FECD donor, age gender matched, and blood from the patient, and also from age gender match control. I supply the clinical examination, specular microscopy, all these features, and try to analyze the first degree relative, try to analyze the pedigree. We later, after our few observation, we later included the spouses of all the family. So far we have sent so many samples in Niger, and already 55 family sample have been sent. 2 PhD have been completed there, 2 PhD are ongoing by their student under this lady Devoshmita. This is the CTG 18-1 chromosome, and you see the repeats are like CTG, CTG. So they are on chromosome number 18. So in our series, the findings are surprising. Only that 7% of Bengali population, FECD population, is having TCF4 CTG 18.1, whereas if you compare with the control, it is 2%. So it was not statistically significant, indicating the other kind of genes or genetic mutation are responsible. So further studies are going on. If you see parallelly the published data, it is from, like in India lowest was 17%, but on higher side from British, Caucasian or American or you say German, they're all nearer to 80%. This is a typical family of where CTG repeat. If you see that more than 100 repeats in all the proband is also 130 repeats, the other are 113, 114 repeats. We have all 3 generation, and I have given the second generation, they are FECD positive patient. Then we look for the other genes. So we have published those data, and there is one called LAMC1. These are all like laminin subunit gamma unit 1. There are only 1 or 2 studies are available in the literature. We said that it is positive for our patient. Then we have also studied that ATP1B1 expression in FECD patient. These are all like their language, not my language. So it is also important because this particular gene is maintaining the sodium and potassium across the cell membrane, sodium potassium pump mechanism. Then we have also studied PGS1 polymorphism in FECD. This is also manuscript submitted. Recently we published this last month only, we have published another kind of, and we have given a hypothesis that it is different kind of gene is also acting in our patient population. So I am giving some example where you see that they are FECD positive, but all the repeats are less than 50, like 31, 32, 31. These all 3 are positive, and I have been operated 1 patient. Spouses are not included in this. But this is another family where we have studied the LAMC and PGS1. They are also positive, but CTG first one is all are below 50. Then this is a strange family. If you see that I have only studied 2 generations, there are many family members. I have yet to go to them and take all the samples, at least blood sample from this. Among 11 people, 9 of them are FECD positive, and all checked by clinical examination and specular microscope. Some of them very old, they cannot come, I have to go to them. So summary, unlike Caucasian or other Western world ethnicity, TCF4 CTG18-1 expression has little role in FECD in Bengal. Probably it is multigenic origin in Bengal. Environmental or other epigenetic factors may have a major role in our region. Very difficult to study them long way because it is very expensive study. Then, so what is the significance of FECD genetics? Clinical significance right now, not bearing any clinical decision making, but of course we can predict the possibility of future FECD in younger individual, explanation about incidence and inheritance pattern, future direction, correlate genotype and phenotype over a long duration, predict progressive versus some FECD we know that it's never progressive, it remains like this. We can also explain some FECD, they remain static. Why they are static, we can explain that. Identify candidate for gene therapy. At all we find certain gene, it is not a single gene, because in Caucasian they have tried to edit the TCF4 because 88% or 80% of their FECD patients. So they are, for them it is easier, but for us it is very difficult. May answer questions about high prevalence in West Bengal. Thank you very much for your patience hearing. Any question? Jerry?
There are only two people in this hall who have enjoyed completely the talk. There's Jerry and Summer. The others are learning. Yeah, but it's always fascinating to have such talks coming in because this is the future, and we don't want to be caught unawares later with this gene therapies coming in a big way in so many diseases. So it's nice that we have some of our clinical people taking an interest in this.
No,
Thank you,
because
Summer.
I was discussing with you. This is my next 5-year project. I have applied for PhD in some of the institution. Unless I am being disciplined, I could not do the whole study. So somebody must make me disciplined because it is just haphazard study in between clinical. So if I get a right protocol, right thing, so I have applied.
Importantly, a right monetary grant, because these gene research is not cheap. It goes into several lakhs and even crores.
Right.
If you have to study a large population, say 100 plus, you're spending about
You
a
have to
crore of rupees.
collaborate with someone.
So you need grants from the government and
yes,
from,
yes, that's what we are doing now.
more power to you.